So, without further ado, Sanjay Mathew is going to talk to us about EEG gamma oscillations as a prognostic marker for IV ketamine in TRD. And Sanjay's one of the doyens of our field and hails now from Baylor. Thank you.
Thank you, Rupert. Thank you to Sarah and Rupert for organizing this and for the invitation to present. Let me see if it's coming online here. Okay, great. So, so thanks again, and this will actually touch on some of the points that Marco was making before, so that was a nice setup for my talk. And I'll be presenting some data that's been published, as well as talking about a new study that's about to be launched that's funded by NIH. These are my disclosures. The work I'll be studying is funded by VA and NIH. And we'll be talking about the background of EEG and why gamma in particular is of interest. Talking about a pharmaco-EEG study that we did of IV ketamine in late life TRD, and then future directions in future studies. So why are we interested in integrating EEG with clinical care for the use of ketamine? If we had good clinical biomarkers, maybe it wouldn't be necessary. And many of you in the audience have been focused on these biomarkers clinically that could be potentially predictive of response. And these are some of them, including baseline clinical features, family history of alcohol use disorder, higher BMI, as talked about this morning, childhood trauma could be implicated, anhedonia, anxious features. Post-treatment, some things that have been looked at include ketamine-induced dissociation, visual analog happiness response, and then blood pressure elevations. But by and large, these have not been large-scale replications such that a patient comes in pre-baseline or post-ketamine. It's not going to be a definitive way of identifying who's going to respond and who's not going to respond. Of course, our holy grail is developing brain-based biomarkers, including any number of imaging modalities, fMRI, PET, EEG, spectroscopy, for the ultimate goal of patient stratification and prediction. So one of the more recent predictor papers and moderators, in fact, was pooling, this is work by Rebecca Price, and several of you in the audience contributed data for this pooled analysis. This is an analysis of 800 plus patients across 17 studies of IV ketamine in depression. And unfortunately, nothing really came out strongly as a predictor or moderator of ketamine versus midazolam or saline response. There was a suggestion that patients who are more treatment resistant experience larger efficacy with ketamine versus placebo, but no biological measures, including BDNF, came out positive. So Jonathan's brother, Dan Javitt, wrote a nice review looking at ketamine-induced biomarkers for glutamate-related development. And most of the work has been in fMRI bold response, as well as several studies in spectroscopy looking at GABA glutamate response to ketamine. Now, a recent review by Medeiros looked at more than 1,700 patients, 69 studies, and tried to identify what are the most consistent brain-based biomarkers or predictors of response to ketamine. The first was increases in functional connectivity within the PFC. The second was increases in activation within the striatum, and this was using arterial spin labeling or task-based fMRI. And the third finding was increased gamma power in frontoparietal regions, which is really the topic for the rest of the talk. Gamma power was assessed by both EEG and MEG. There were 3 independent samples, including one of our own, showing this effect with one negative report, Macmillan, in 2020. So when you look across multiple studies, it appears gamma power seems to be a relatively well-replicated marker associated with response to ketamine. So why EEG? And this is just an example of the setup. In contrast to fMRI, low-cost, portable, combination with other methods, of course. We get into issues with fMRI, of course, with patients who are obese and exclusion for motion artifact and so on. And obviously, this is something that can be taken to the real-world clinic in busy clinics, and hopefully there are not too many exclusion factors. So the resting state EEG measures the dynamics of spontaneous patterns of synaptic communication. So essentially, it produces a mixed signal that reflects the patterns of synchronized firing by clusters of pyramidal neurons in the neocortex. And using a Fourier transform, you can estimate the oscillation content of the signal, which is the contribution of different firing rates to the overall signal. And you can see the different frequencies. Last talk, Marco was talking about alpha decrements as well as changes in theta. We're focusing here on the high frequency gamma, which is generally defined as 31 plus hertz. And when no task is present, you can take this spontaneous activity to represent the functional resting state of the brain. And we do this in an eyes open, eyes closed paradigm. So one of the interesting things about the EEG neurooscillatory measures are its implications back to understanding cortical microcircuitry. And this is a schematic published by Clinton a couple years ago, looking at the interplay between GABAergic interneuron inhibition of pyramidal cell glutamatergic output. So in a baseline non-stress state, you can see somatostatin inhibition of glutamatergic neurons, parvalbumin containing interneurons inhibition, and you can see the output in a healthy state is robust. In a chronic stress state, you have enhanced function of parvalbumin inhibition of pyramidal neuron output, diminishing output, as well as somatostatin diminished function, which leads to associated with dendritic atrophy. So a schematic trying to make sense of how gamma oscillations can interplay with this is that gamma activity, the high frequency activity, is thought to reflect the increased excitatory drive to specific cortical regions, which then trigger rhythmic feedback mechanisms via these local fast spiking interneurons. And you can see this motif here, feedback inhibition in the high frequency and feed forward inhibition. It was mentioned earlier this morning about what is the role of LTP with ketamine's effect. One interesting aspect is high frequency oscillations have parallels to activity dependent plasticity mechanisms such as LTP. And so one hypothesis is that potentiation of gamma band activity could be a potential biomarker for local microcircuit neuronal integrity. Also, although these are scalp electrodes, we are making hypotheses at the microcircuit level. And so these, of course, work derived from animal models with EEG. So ultimately, what are you trying to model when you're looking at gamma band activity? One of the models, of course, that has been discussed at this meeting is ketamine ultimately leads to disinhibition and a glutamatergic surge, which then triggers downstream neuroplasticity cascades, including AMPA receptor potentiation, TrkB, et cetera, due to its initial blockade of NMDA channels on GABAergic interneurons. And so animal and computational models have showed that NMDA inhibition leads to suppression of these GABAergic interneuron. Disinhibition increases the synchrony of firing patterns, and this leads to a presumptive glutamate surge. Disinhibition can be inferred clinically in the EEG through increases in gamma band power, and the peak gamma power seems to correspond to peak PK. Now, of course, this is not specific to ketamine, but perhaps one of the best early demonstrations of this was by Jerry Sanacora about a decade ago now, in which healthy volunteers were given 2 doses of the low trapping NMDA selective antagonist. This is lanisamine. This was a drug by AstraZeneca at the time, which was studied in TRD, which ultimately failed in later stage testing. But what's shown here is when you give ketamine to healthy volunteers, you see a dramatic surge in the gamma band activity. You also see this in the high dose of lanisamine and not so much in the lower dose of lanisamine, and really no effect in placebo, which very much mimics the PK of this drug. And so work that we did, this is led by Marijn Leifheit and Nicholas Murphy in our group, we added on EEG to a dose identification study in late life depression. And the goal of the trial was to essentially, in a efficient manner, using a Bayesian adaptive randomization scheme, try to identify quickly the best dose among 0.1, 0.25, 0.5, and then midazolam as the control. We did baseline EEG, we did EEG during the infusion, 24 hours later and 7 days later, and carried forward a variety of measures. The Bayesian adaptive randomization scheme essentially was playing the winner and pruning the loser doses, such that if you just did this in a frequentist parallel traditional design, you'd need a sample size of about 100 patients, like the FAVA study that Jonathan talked about yesterday, to try to identify your best dose. Turned out we were able to identify ketamine 0.5 milligram per kilogram after 33 patients, pruning the 0.1 and the 0.25 dose rather efficiently. The statistician was Chuck Green, who's a Bayesian wizard, and so convinced us to do it in this manner. And the VA was happy because we saved them a couple of years on the grant. And so we did find 0.5 was the most effective dose. Midazolam, interestingly, had a 46% probability of a response, so it was not a negligible response, midazolam, for a single dose through day 7, which was the primary endpoint. And so the findings pretty much mimic what was found in FAVA with the 0.5 milligram dose. Now, all of this is to set up the EEG finding, which was when you compare the ketamine versus midazolam, so similar to what Jerry had found and other studies, there was a dramatic increase consistent with the time course of the ketamine infusion during the 40-minute infusion in the ketamine arm. This is increases in gamma frontal power, no effect in midazolam, and no effects later on. When you look at the relationship between clinical outcome, this graphic shows there was a relationship between the percent delta in the frontal gamma and the percent change in the depression rating scales over the course of the 7 days, with essentially no relationship in the midazolam arm. So there was a relationship between this glutamate surge, or the gamma, I should say, the gamma surge, and outcomes in this model. Now, of course, this was a highly experimental study. Patients were excluded from all kinds of comorbidities. Nothing to do, if you would, with what you see in the real world. Patients on multiple medications, not being washed off. They're not at a GCRC or a clinical research unit. So can you test this model in a real world, busy ketamine clinic in a community setting? And that's what our study will be testing. And this is work by Brittany O'Brien in our group. We've been collaborating with this clinic, modeling the outcomes over time. And as many groups have demonstrated, you see some groups of patients who have very rapid, robust responses to 6 infusions of ketamine over 3-4 weeks. You have a group that doesn't respond at all, and then you have a group with more modest baseline severity who have a slower response. So the model we'll be testing is adapted from Alison Nugent's initial work looking at the relationship between ketamine and EEG, where we're hoping to identify a sweet spot such there's an interaction between baseline gamma power and how ketamine will induce gamma power, such that we hypothesize good symptom reduction in this quadrant with ketamine will induce gamma potentiation in a setting of low baseline gamma power, while the other permutations of this would have perhaps less response or suboptimal response. Her study was done based on single infusions, so we're testing this in the clinic setting at baseline, where patients will have a fixed order saline EEG session followed by a ketamine EEG session at infusion 1, but then also at the midpoint of their 8 infusion treatment trial, they'll get another EEG to see, first of all, is this a biomarker that's stable, and does it change as a function of clinical care, clinical improvement over 3 infusions? How useful is this for demonstrating persistence of benefit, and so on? So the paradigm is repeated at midpoint, and we hypothesize that, relative to saline, there will be not a signal for gamma, and the baseline corrected signal during the ketamine infusion, we hypothesize at 60 and 90 minutes will be elevated. So besides the resting gamma effects, we'll also be looking at a measure of evoked gamma. And one of the measures that you can do for evoked gamma is something called auditory steady state response, which is the response to an auditory click train of 1,000 millisecond duration. And what you see with this ERP is a 40 hertz entrainment of cortical activity, which is phase locked to when the tone onsets. ASSR has been used in a validated measure in animal models and human models of NMDA function. There's shown a direct relationship between channel occupancy and gamma power. The output and the interpretation of the output represents the integrity of sustained inhibitory interneuron firing, specifically within auditory cortex in this case, and it's a proxy measure of NMDA receptor gamma oscillations. So we will be getting the resting as well as hopefully confirmatory evidence from ASSR. And in a study we did several years ago, led by Nicholas Murphy as well, we looked at a oral NMDA antagonist. This is a glycine site NMDA oral drug. This was a drug that Vistagen had, and we looked crossover design in healthies, and essentially what we found was a dose response relationship in increasing 40 hertz ASSR power, such that the high dose, which was believed to be the therapeutically effective dose, had elevations in the ASSR power and also in phase coherence. And I will not be talking about neural complexity markers, just to mention that the EEG gamma, of course, is a linear measure, and there's been a lot of interest in nonlinear measures of neural complexity, including something called LPZ, Lempel-Ziv complexity, as well as multi-scale entropy. And these are measures you can obtain through the standard EEG. And so we'll be looking at this as additional sort of proxies, and as recently published. So areas of uncertainty. Are these ultimately, the EEG measures and gamma oscillations, which is the focus of this talk, are they correlational? Are they causative? What is really the biological significance of this? And we do need to distinguish, although that meta-analysis suggested this is a well-replicated finding, there are certainly negative findings. There have not been enough studies comparing TRD samples to MDD samples versus healthies. So our study will have 3 arms to be able to make those comparisons, and also a remitted depression arm to understand state versus trait measures. There's been a lot of interest in cross-frequency coupling analyses, looking at the relationship across frequency bands, including theta gamma coupling, which has been implicated in information processing and signal integration. And what is the relevance for this ketamine-induced gamma band activation across psychopathology, across the disorders that we treat? We're restricting this sample to MDD, although in real-world ketamine clinics, of course, this kind of work has to be extended if there's a positive signal. And then ultimately, we want to be able to, in a TRD referral center, where we have all of the modalities, VNS, TMS, DBS, et cetera, how do you selectively and precisely push a patient? And can you use ketamine-induced gamma band activity as a relatively low-invasive, relatively simple way to aid in precision therapeutics? And ultimately, I mean, we will be combining this with fMRI measures, and then we were recently, we've collaborated with a group in Yokohama, Japan, who have developed a AMPA receptor PET ligand. It's called K2. It's a C11 PET ligand, and they showed that in epilepsy patients, they showed alterations in AMPA PET distribution in these patients, and we're collaborating with them to do this work in conjunction with EEG. And so hopefully I can show that data in 2 years. Thank you.
Wonderful, Sanjay. Thank you for giving us time for questions and for bringing us up to time. So I'm going to take a question here first.
Thank you, Sanjay, for this very good talk. I wanted to ask you whether doing this kind of EEG studies would be relevant at the early stage of development of a new NMDA receptor antagonist by comparing it with ketamine and for selecting the dose, for instance.
So can you speak up, please? So the question is, how relevant is it at an early stage of drug development? Is that right?
Yeah.
Okay.
I mean, I wish I could say that definitely things like ASSR, which clearly separated the high dose of the Vistagen compound from the low dose. Unfortunately, it didn't have any implications for performance in the phase 2 clinical testing, which was negative. So it engaged the target, but it didn't have any read-through clinically. It is, in my view, a good way for dose sort of separation type studies in trying to go forward with a dose that clearly has target engagement properties that you can confidently go into phase 2 testing with that information.
Okay.
And now, which is better for that purposes, ASSR versus resting gamma, that's not as clear. And the other example of negative phase 1 is Jerry's own data. Unfortunately, the implications, it's not like the drug didn't work, it was just a very high placebo response.
That's what I was going to,
Jerry
I mean,
can
it's
speak to
difficult to say because that's exactly what was used for linistamine, and it looked like it predicted. If you look at the first phase 2 trial, it hit on every primary, every secondary. In the second study, the drug effect replicated identically. The only thing was the placebo effect went from there to there. So, the question is, did it predict a good dose and the first study was right, or was the second study right? But I think it's hard. Between the two of those, I don't think you can rely on it. I think that's what you're saying.
Yeah, and I would also add that in Jerry's healthy volunteer, the 150 dose, which mimicked the ketamine response on the gamma band, was not used in the ultimate clinical trials. They ended up using a 50 and 100. So maybe the company wanted to err on the side, maybe you could speak to that, err on the side of no dissociation. That's why they didn't go with the 150, but it leaves you wondering if they had gone with the 150, would that have been a more robust response?
So it definitely was the fact. It was complicated. It was for other reasons too. They had to keep it down. I do think you may be touching on the core because there wasn't much dissociation. So the question was, there was also there was good blinding, so there wasn't functional unblinding, which may have played a role. But the interesting thing was that there was evidence of the lower dose actually doing a little bit better. There was some evidence that the lower dose actually did better than the higher dose, and that was part of the reason for the second study. They lowered it even.
Okay, I think perhaps you could retain your question for the interval, but I'd just like to ask Marco to get a youngster in with his question, if that's all right.
Yeah, sure thing. I mean, thanks for the talks. It was really interesting to see and really excited to see some more of this work. I had two quick ones. First one was, in your dose finding, was there a reason why you didn't go much above 0.5 milligrams per kilogram? And if you did, do you think that there's also this kind of saturation effect where the gamma wouldn't increase past a certain point, and that could be interesting for the kind of personalized medicine? Perhaps we could start.
Yeah, the reason we didn't go above, this was a older population, so we were a bit more concerned about a 0.75 dose, although that would have been nice for the modeling to be able to show that.
And just to tie in with some of the earlier talks, do you think that this could also allow us to pinpoint the opioid story? In that, do you think the naltrexone would reverse some of these gamma effects potentially? Because I know you focused on NMDA, but actually it could be interesting to see.
Yeah, no, that would be a fantastic experiment to look at the naltrexone effect on gamma potentiation. And we will at least get, since this is a real-world clinic, we will have some patients who come in on naltrexone for the treatment of opioid use disorder. We're not excluding those patients. So for what it's worth, we'll have a subgroup.
I'm really keen that that's fantastic. Thank you very much indeed. I'm really keen not to keep you from your lunch too long.